Okay, can I go? Okay, so we are back, and we are very honored to have with us Professor Jack Dekkers, who actually doesn't need introduction, but I will try anyway. Jack has been professor of animal breeding at Iowa State University between 2002 and 2014, and since 2014 is Curtis Distinguished Professor in the Department of Animal Science, where he is also a section leader. Jack focuses his research on quantitative and statistical genetics, including the integration of quantitative and molecular genetics and genomics for QTL mapping, genome-wide association studies, and genome prediction and selection. And today he gives us a very interesting talk on the genetics and genomics of potential indicator traits for disease resilience of growth finishing pigs. Thank you very much, Jack, and I give you the floor.
Okay, thank you very much, Elisabetta, and yeah, thanks also for the invitation. It is indeed an honor to talk to this group. No, it's a keynote. It's called a keynote talk, and I think in part because it's not about FANG. This talk, although we are using some gene annotation information in some of our analyses, but hopefully it will still be of interest to many of you. Okay, so the talk is about resilience. Now, we know that when it comes to infectious diseases, resistance to some pathogens is controlled by a single gene, and the F18 E. coli mutation in that gene is quite famous for that. The mutation removes a receptor that in the intestinal wall that the bacteria uses to attach to the intestinal wall, so that the receptor isn't there anymore, or not functional anymore. Then the bacteria has no way to attach to the inner lining. But when we talk about other pathogens, most pathogens, resistance or susceptibility to most pathogens is highly polygenic, and maybe there are some genes with larger effects, and we've certainly seen that with infections or susceptibility and resistance to PRRS in pigs. But generally, it's very polygenic. Now, of course, there are examples where gene editing has been successfully used to introduce complete resistance of animals to a pathogen, and the PRRS mutation or the PRRS edit in the CD163 gene is a good example of that. We're here focusing on using existing genetic variation rather than creating new variation by gene editing. Not that that technology isn't very promising, it is, but is it going to work for all the other pathogens that are out there, where there may not be single or a limited number of genes that can be edited to make the animals resistant. Also, the pathogen is evolving, and there are new pathogens on the horizon, well, that we don't even know yet about yet. So for that reason, we have focused on disease resilience, which is the ability of an animal to be minimally affected by disease or to rapidly return to the state before the disease. So basically the ability to cope with disease, given that we're not going to be able to eliminate all pathogens from the environment, or make animals completely resistant to all pathogens. And that's, of course, complicated by the breeding structure that we have in livestock species. Here, an example in pigs, where the selection and most of the data recording takes place in nucleus herds that have to be maintained with high biosecurity. And that, of course, makes it very difficult to collect data related to disease resilience. So to address this, I've been fortunate enough to be a part of a large consortium led by Mike Dick and out of University of Alberta, and Graham Plastow has been very involved. Graham, I believe, is online. And in 2015, as part of, we obtained funding from Genome Canada and Genome Alberta and Genome Prairie initially to start a large-scale project on disease resilience in grow-finished pigs. Quite a number of institutes and universities involved, as you see here, and also very important, very strong industry component to this project through PigGen Canada. PigGen Canada is a research consortium of pig breeding companies. When we started, it included seven breeding companies that are listed here. Two of them have since dropped out, so we're left with 5 industry partners. And these partners, they provided the animals that we are working with, as well as funding, as well as helped us with directing the research and focusing on implementation. So that started in 2015, and what it involved was at a test station in Quebec, a natural challenge was set up, which was initially in a barn that included a nursery and a finisher. Pigs from neighboring farms with health problems, known health problems, were brought in to seed the barn with a host of pathogens, and then to maintain that through a continuous flow system. So every 3 weeks, a new batch of pigs from one of the 7 industry partners was entered into this natural challenge facility, where they had contact with the older batch that was already infected, and that's how the infection pressure was maintained. Before these pigs went into the natural challenge facility, they went into a quarantine nursery, and where we took a lot of samples, and that's a lot of what I'll talk about is what we collected there and how we analyzed that. So in total, so this ran for about 7 years. In total, about 4,700 Yorkshire landrace weaner pigs. They were barrows from clean multiplier farms from the 7 companies. They were entered over the 7-year period. So the pigs, they were born in clean multiplier herds. Barrows are sort of a byproduct of the maternal line crossbred program of these breeding companies, and so they were transported to actually what I'll show here. The test station was just north of the St. Lawrence River. US is here, Quebec is here. And they first entered into a quarantine nursery, and then about 19 days later, they were transferred to this challenge facility, which consisted of a nursery and a finisher in the same airspace. So this is where they were challenged, and eventually, if they made it, they were sent to market, and we also got slaughter data. So there are lots of pathogens in this barn, and it wasn't consistent from time period to time period. Sometimes some pathogen disappeared from the barn, and it had to be reintroduced. So quite a number of viruses. Initially, we also had PCV2, and the pigs were not vaccinated for PCV2, but because the challenge was a bit too severe, fairly early on, vaccination of pigs for PCV2 was implemented before they got into the facility. And number of bacteria and some parasites. And the weekly mortality rates you see here for part of this 7-year period, it fluctuates a lot. And there was a very dedicated veterinary team that kept very close eye on making sure that it didn't get out of hand, but also to make sure that we maintained disease pressure. So different strategies were used to mitigate or to maintain more or less level a certain amount of disease pressure. But obviously, it's a very dynamic system. So under the challenge, we collected growth performance. We have daily feed intake, also daily water intake. morbidity in terms of health scores, mortality, and all the veterinary treatments were recorded. So these are what we call our resilience phenotypes. They were collected on all 4,700 of pigs. And they were all genotyped using a high-density SNP panel. This is an example of one of the batches. The red ones are the ones that died. Reasons of death, quite a range, but a lot of intestinal problems and respiratory problems, reflecting the pathogens that were in the barn. And the number of individual health treatments, there were some group treatments, but then the individual health treatments, some pigs didn't need any treatment, and others required 10 or more. So quite a bit of variation there also that we saw. The first question we had, well, given that this is not a controlled disease, it's very dynamic from batch to batch. Is the data we're going to get out of this even heritable? And so Austin Putz, who was a PhD student at the time, now works for Hypor and Hendrix Genetics. He analyzed the initial data and found that, it's quite heritable. So this average daily gain in the challenge nursery and in the finisher, very respectable heritability, similar to what you would see in a nucleus herd. Also the same for feed intake and FCR, same for carcass traits, and also the ones where we're mostly interested in, number of treatments per day and mortality in the challenge nursery and then across the nursery in the finisher. No, low heritability, but higher than what we would typically see in literature, where the incidence of disease would typically be quite a bit lower. So again, keep in mind that these are multiple diseases that animals are treated for, and so we lump everything together, and the same with mortality, and yet we obtain fairly reasonable heritabilities. Another PhD student analyzed a larger data set and also finds reasonable heritabilities, and this has all been published. And he also looked at drinking behavior, and also we found reasonable heritability there. And then he also looked at reaction norms, where he quantified the disease pressure, and then looked at G by E, and found some evidence of G by E depending on the level of disease pressure, which, as I mentioned, fluctuated over time. He also did a GWAS, and this has all been published. I'm just going to show you the GWAS for average daily gain in the challenge nursery. So that's where they're mostly affected. And we find quite a strong signal on MHC. Now, when you look at the QTL database, and you look at growth rate QTL, there's very few reports of MHC showing a QTL for growth rate. And that's because most of these studies are done in healthy environment. And you can see here that under challenge, MHC does become important for growth rate also under the challenge. One of the main reasons for doing this study is to develop these early indicators of resilience that potentially could be measured at the nucleus level where there's no disease. And so that was I'll talk most about that for the rest of this talk. So in the quarantine nursery, quite a number of indicator traits were investigated. And the requirements we have for a trait to be a good indicator trait for genetic selection is that it has to be heritable, and it also has to be genetically correlated to the resilience phenotypes. And we look at CBC, natural antibodies, high immune response, in vitro immune tests, phagocytosis test, and the blood transcriptome, as well as the proteome, metabolome, microbiome. And then finally, I'll talk about stress hormones in hair. And all of those were collected in the quarantine nursery prior to the animals getting into the challenge. Now, CBC, I'm not going to talk about. Shoshun Bai, a PhD student with Graham Plastow, worked on that, and that was published. I also not going to talk about the blood proteome that's also been published, nor about the blood metabolome. Elda Dervishi, a postdoc in Graham's lab, worked on that, and that has been published. Some very interesting results, but I just don't have the time to cover all of that. What I am going to talk about are the ones that are in red. So the first one is natural antibodies. Yulu Chen, a PhD student in my group, worked on that. Natural antibodies are present even in the absence of pathogens, and they bind to conserved antigens across pathogens. We looked at 4 of them, 4 of these mitogens, and or antibodies, natural antibodies to 4 of those mitogens, and that was all done on blood that was collected in the quarantine nursery. So again, before they entered the challenge. And she found reasonable heritabilities. She looked at 2 isotopes, IgG and IgM, natural antibodies to those antigens. IgG was lowly heritable. IgM was quite reasonably heritable. And IgG, we also had quite a large maternal effect, which is not surprising since IgG reaches the piglets through the colostrum, and they're only about 30 days old by the time these blood samples are taken. So in terms of genetic correlations with resilience, and this gets a little complicated, but just want you to focus on the trends. So this is the IgG and natural antibodies and their correlations with mortality in the nursery, in the challenge nursery, in the finisher, number of treatments, and also average daily gain in the quarantine nursery and in and under challenge, as well as feed intake. Here you see that KLH IgG and LPS IgG are positively correlated genetically with growth rate under challenge in the challenge nursery when they're first exposed to the pathogens. And there so the interpretation there is that higher levels of antibodies to those antigens, IgG antibodies, is genetically correlated with higher growth rate, so giving some protection or being associated with some protection. Standard errors are large, and that's one thing you'll see throughout these immune assays. We were not able to do on large numbers of animals, so typically about 1,000, a little under 1,000. And we just looked at lots of immune assays to identify ones that are promising that then can be further investigated. So also for mortality, you see negative genetic correlations, so higher level of that natural antibody genetically associated with lower mortality, but large standard errors. When we look at IgM, higher heritabilities, and most of these tend to be negative, but again, no large negative correlations and large standard errors. But there is some indication that these natural antibodies could be indicators upon further validation. So especially IgM, because the higher heritabilities, but genetic correlations definitely need more validation before that can be implemented. The other technology what we looked at was the high immune response technology, patented technology from Bonnie Millard at the University of Guelph that has been implemented in cattle, in dairy cattle, and also beef cattle. So she measures cell-mediated immune response and antibody-mediated immune response in dairy cattle. She finds heritability of about 30%. The cell-mediated is like a TB test where you inject an antigen and then you measure the skin thickness. I think 1 or 2 weeks later, and this is an ELISA test. And she has shown that these tests are genetically correlated with disease resistance, with disease incidence, and has been implemented by CMAX Canada as Immunity Plus. And so when we started this project, we approached, several of us approached Bonnie and see, well, can you also implement this in pigs? And so that was done. So Julie Schmidt, who is a postdoc in her lab, Bonnie's lab, so worked with the staff at CDPQ at the facility to implement these both tests. And here you can see that and it's injected in the flank, and the other one gets a placebo, the other side, and then they measure fin thickness 1 or 2 weeks later. The heritability, so Mark Cheng, who was a PhD student with me, analyzed this data. He finds reasonable heritabilities, a little bit lower than they find in dairy cattle, and some litter effect for the antibody-mediated response. The genetic correlation between the two was slightly positive, so that there is some genetic correlation between these two arms of the immune system. What we are, of course, interested in is, well, if we measure that in the quarantine nursery, AMER and CMER, is it genetically correlated with subsequent disease resilience? So here you see growth rate in the quarantine nursery, in the challenge nursery, and in the finisher, low correlations. They're a little bit larger when we look at the number of health treatments that the animals receive in the nursery. And also for mortality, they're a little bit larger, but large standard errors. What surprised us is that for CMER, we found an opposite direction of these genetic correlations than what we would have expected, and what's seen in dairy cattle. In dairy cattle, higher AMER and higher CMER is associated with less disease. And here we find that also for AMER, but not for CMER. And this may indicate that the pigs, although they came from clean multiplier farms, they may have been exposed to some minor pathogens when they showed a response that when we take the samples in the quarantine nursery. And we'll see some evidence of that coming back later on also. Mark also did a GWAS and found a very strong signal for MHC on MHC for AMER, but not for CMER. For CMER, it was very, very polygenic. The next immune assay that we looked at was proliferation of PBMCs from young healthy pigs. And again, those were done on blood that was taken at the quarantine nursery. The work was done at McGill University, Caroline Gilbert, and it's been published. And so we looked at a total of almost 900 pigs and exposed the PBMCs to 5 mitogens that are listed here, and then computed the blastogenic index score at 3 time points after stimulation of the PBMCs. And that's the results I'll show you. So heritabilities, so the 5 mitogens here, heritabilities at the different time points, and then also area under the curve, reasonable for CON-A and PHA, and also for PMA, but not for PWM and LPS, surprisingly. So in terms of genetic correlations with resilience, we only looked at the ones that are indicated, well, the 3 that showed reasonable heritabilities. So those and these results are also all published. The genetic correlations of response to these mitogens at the 3 time points with the number of health treatments in a challenge nursery, and given the large standard errors, it's most important just to look at the overall trends across time and across different resilience traits. And here they tended to be negative. So higher response to of the PBMCs to these mitogens, fewer treatments genetically. When we look at treatments across the challenge nursery and the finisher, correlations get a little bit lower, but still that same tendency. Mortality, also, even larger standard errors, but a tendency for the correlations to be negative, so less death, lower mortality, lower risk of mortality of pigs that genetically have higher response to these mitogens. Mortality in the finisher and across the nursery in the finisher. We also did health scores. So scoring pigs, every pig is scored on a scale from 1 to 5, with 5 being more healthy. And all those correlations, again, huge standard errors, but all those correlations were quite large negative. Most of them were quite large in both the challenge nursery and the health score in the finisher. Growth rate tended to be positive. So pigs that had a better response, a better immune response, PBMCs responding higher, tended to have higher growth rate in the challenge nursery. And in the finisher was a little bit less clear. So, some interesting results that came out of this, but again, they need further validation to deal with the large standard errors. Next, we looked at immune stimulation assays on blood of young healthy pigs. So in contrast to the PBMCs, that needs to be done on fresh blood, isolating the PBMCs and stimulating them. These assays can be done on stored blood, and this work came from Claire Rogel-Gaillard and Fanny Blanc at INRAE. And also was actually most of the assays were done in Graham Plastow's lab. Fishesh and Shushun Bai analyzed the data. Shushun was a PhD student with Graham and now works for Turkey Hybrid, Hendricks, and Fishesh Bhatia is still a PhD student with me. And so they looked at a total of over 1,500 of these pigs, again, blood taken in the quarantine nursery. And they looked at both the concentration and the fluorescence activity of these 9 cytokines. And then under 3 conditions, non-stimulated blood and blood stimulated with LPS and a combination of PMA and ionomycin. Heritabilities were tended to be low for the non-stimulated assays, blood without stimulation, so the measurement of those cytokines in unstimulated blood. But with stimulation, especially LPS stimulation, we got slightly higher heritabilities. So that definitely was promising. In terms of genetic correlations, and this was only done for the more heritable cytokine levels and assays. And here I'm showing growth rate in the quarantine nursery, so without challenge and under challenge. So generally, these correlations are negative, indicating that pigs whose bloods produced more cytokines after LPS stimulation had lower growth rate in both the quarantine nursery and under challenge, which is maybe opposite to what we expect. I'm not quite sure how we can explain that, but that's something that we still are trying to investigate. And to some degree, we see the same for the PI stimulation and a little bit even in the non-stimulated. Okay. When we look at genetic correlations with treatment and mortality, they tended to be positive. If you look at the red and the gold, or the cardinal and gold, which is the treatment and mortality under challenge, where you see most of the disease, they tended to be positive. So again, opposite direction to what we maybe expect. They also looked at a phagocytosis test, which can also be done on blood samples, and same group of people involved. This was done on almost 1,000 animals. Again, assays done on blood taken at the quarantine nursery, looking at 5 different cell types and both the cell count, the percent of those cells that phagocytize, and their fluorescence activity, so how much they phagocytize. Heritabilities were reasonable, up to 40% or 50%. The cell counts, but also the phagocytizing percent, fluorescence a little bit less heritable. Genetic correlations, we focus on the red one, that's growth rate under challenge. Fairly low for cell count, and Ashu Shunbai has a separate article where she showed that also, that the cell count measured on healthy animals isn't very predictive genetically of disease. Percent phagocytizing ability there was some evidence, but again, opposite direction to what we expect. So pigs that have a higher capacity for phagocytizing genetically have lower growth rate under challenge, although that did recover in the finisher. And also for fluorescence, well, for fluorescence, it was a little bit less clear. There was something going on in the quarantine nursery with large standard error. So also that needs more investigation. And then with treatment and mortality, if you look at treatment, the cardinal color here, tendency for, well, this is with cell count, with phagocytizing ability. So here the cell count, higher granulocyte count, a tendency for genetically more treatments, but fewer in the for phagocytizing ability. Here we do see the expected direction, that higher phagocytizing ability, tendency for fewer treatments and less mortality. So, and especially the eosinophils are interesting here, although I understand that the eosinophils don't have a huge phagocytizing capacity, but we did find that negative genetic correlation in a favorable direction for both phagocytizing ability and fluorescence of eosinophils. Then we also looked at the blood transcriptome. So again, those were taken in the quarantine nursery. We used the QuantSeq or 3' RNA-seq with a globin block to analyze, to get gene expression on 1600, and we actually now have it on 2500. There's Kyu-Sung Lim, who was a postdoc in the lab, is now a faculty member in South Korea. He did the initial analysis at the phenotypic level between gene expression and the resilience phenotype. So is gene expression level measured on blood, is it predictive of disease resilience phenotypically? But here what I'll talk about is the genetic analysis that he also did, and that is about to be accepted by GSE. So he estimated heritabilities of all 14,000, the expression of all 14,000 genes in blood of these about 1,600 animals. And he did it with or without correction for CBC, for cell counts, because dealing with the fact that blood is a mixture of cells, and to account for cell composition. Generally, heritabilities with and without, they tended to be very similar. So not a huge effect of cell composition on heritability estimates. But for litter effects, we did find, and it's kind of expected, that expression of mitochondrial genes had large litter effects, and so also some genes on the X chromosome. And of course, we do an analysis where we don't account for mitochondrial DNA or the sex chromosome. So that's kind of what you would expect to see. And then you look at genetic correlations with resilience. All the different traits here in the different phases of the challenge, and the number of significant genes, depending on whether it was with or without correction for CBC, or was significant for both analyses. So we estimated all these genetic correlations, and significance at 0.05, but then when you correct for multiple testing, then FDR, there are very few genes that have an FDR less than 0.2. So still, even though we have 1,600 animals, it's still a limited data set when you want to get a genetic correlations, which is what we need. So what he did is he did a GSEA, gene set enrichment analysis on the genetic correlations. So here, either without or with correction for the CBC, all the traits on the different phases, and then looked at the genetic correlations of all the genes, expressed genes in blood with these traits. And then it did a gene set enrichment analysis based on Go terms. And he did find that there are multiple immune terms that come up that when you look at the genetic correlations. And the red here indicates that the genetic correlations of genes with those Go terms that were classified with those Go terms tended to be unfavorably correlated with disease resilience. So generally higher level of expression of those genes related to innate immunity, adaptive immunity, or both, tended to be higher expression of those genes tended to be associated with lower resilience. And again, remember, we're looking at a measurement of gene expression in the quarantine nursery when they're still fairly healthy, although this may also be a result of the fact that there may have been some disease pressure already even before they got to the quarantine nursery or in the quarantine nursery, although none of the major pathogens were detected there. So yeah, so when you look at cross genes, there is some evidence of associations between gene expression and disease. Too early to get into any indicator traits or indicator panels that we would develop with gene expression. We need to do more investigation to look into this. As you can see, for a lot of this, we've just only scratched the surface of a lot of the analysis that need to be done with these data. So now we have added another 850. So we have almost 2,500 animals with gene expression data in blood. And now with a grant from NIFA led by Chris Tuggle, we are doing, he's doing a lot of single cell RNA-seq in blood. And now the plan is to do deconvolution of the expression profiles to get expression profiles of individual cell types on all those animals. And that hopefully will tell us a bit more yet. The final thing I'll talk about is something that's also still ongoing, where we're looking at hair. Instead of blood, we're looking at hair. Of course, that's also an easy sample to collect. It's actually cut hair, not pulled hair. Because what happens when your hair grows, hormones that are in the bloodstream are deposited in the actual hair. And so as your hair grows, it reflects the hormone levels that are in the blood at the time of growth. So you could, in principle, take the tip, and that would represent stress hormone levels in blood during the time that part of the hair was grown. No, we're not splitting the hair. We are actually cutting the hair 3 times. The same patch on the back is cut when they just before they enter the challenge nursery. So then we're looking at hair that was grown since birth, basically, and looking at the effects of response to stressors that the animal was exposed to during that time, the stress of farrowing, weaning, being mixed, transportation in this case, changing diet. So a lot of stressors here. And then that same patch is cut twice. So here, where it would mainly reflect disease stress, and here also disease stress and potentially also other stressors. And the idea is that there's lots of stressors that animals are exposed to. In the disease challenge, we're mainly looking at immunological stressors, but all these other stressors we'll see here, and also partially also at the other 2 time points. And the hypothesis is that response to non-infectious stressors are genetically correlated to responses to infectious stressors, and that's one of the things we're getting at. But then, of course, we're also using this to see whether they are genetically correlated with disease resilience. And so that's the data we have so far is just on cortisol and dehydroepiandrosterone taken in hair at the quarantine nursery. And we are also doing the back test was done at that time point, almost 1,000 animals. Heritabilities of the back test were quite good. For cortisol, about 0.2. DHEA was not very heritable. Well, actually, at the zero heritability, it did have some litter effects that you can see here. And then we also looked at the ratio. Genetic correlations of cortisol with the back test. The one reason we did the back test is to investigate, well, is what we're measuring in hair at that time point, is it genetically correlated with a standard stress response test? And we find that those genetic correlations are positive. He also, Fazir has done a GWAS on cortisol, and we find a very strong signal, more than 60% of the genetic variants, that is associated in the window that has the NR3C1 gene, which is a gene that modulates the availability of cortisol and the HPA axis. So this is a very strong candidate that we are looking into further. That's all the work that we've done so far. Of course, now we're also going to get haircuts at the later time points. So we'll be able to correlate them with cortisol and other hormone levels that we are measured at this time point, and also looking at correlations with disease resilience, which is underway. So in conclusion, we think disease resilience is a useful target to improve host response to multiple pathogens and future pathogens that may come along. To do that, we established this natural challenge model. We found heritable variation in growth, feed intake, mortality, morbidity. We did lots of immune assays. Lots of results are still inconclusive. Needs more analysis and more data. The same with the transcriptome, and some encouraging initial results for cortisol. And hopefully we can answer this question also, whether disease resilience parallels resilience to other stressors. So with that, I want to thank all the collaborators, and hopefully I've left some time for questions, but most of you will probably be hungry.
Thank you very much a lot for this talk. We cannot clap because anyway, but we can try.
Thank you.
Okay. Any question for Jack? Not yet? Okay.
Everybody's ready to go eat.
So, I mean, there was an incredible amount of information, and it's also amazing to see the numbers that you have in this study. So you are above 2,000 animals now that you can type and characterize for everything. And I underline that I really was pleased to have Jack to, we didn't really tackle disease in the second part of Gene Switch that we will have tomorrow, but will be really like that this talk is really a kind of a grasp already to the plan, the program that we have tomorrow, where I also hope Jack will join. So one thing that is really interesting is that, if I understood well, I mean, for example, when you look at your immune genes, you have, as a trend, if I understood, you have levels of immune genes that actually are anti-correlated to resilience, correct? I mean,
yeah,
you have high amounts, but in fact, high amounts don't correspond to more protection.
Yeah, we saw that with the cytokines and also phagocytizing ability, and not with the stimulation of the PBMCs. Yeah, what the reason behind that is, it's not clear, and it may be that and it may relate to the response to non-infectious stress also, which we know that immune system is involved in that, because these pigs, when we take most of these samples, they've just been transported across Canada. They're on a mixed. They're on a new diet. Blood samples are taken about 7 days after, and then also some of the assays were done on blood that was taken about 18 days after they arrive. But lots of things going on that may affect what we measure in blood at that time point, and how it could relate to disease resilience. So maybe they are already more stressed at that time point, and we see that in some of these immune assays, and they may be less more susceptible to those stressors as well as to infectious stressors. But that's all speculation at this point.
But you could actually probably test it with new measures, I guess. You could try to modulate it,
Yeah.
And I mean, and I don't know if there are questions. In the meantime, please think of it. Yes. Alan. Okay, so I give the floor to Alan because he's connected with a question with Jack.
Hi, Jack. There's a great talk. There's a sort of long-running dogma that if as an animal you invest in your immune system, that compromises growth. Any evidence from this enormous body of work to support that dogma, or to knock it on the head?
Yeah, we haven't. That's a good question. We haven't looked at directly at that, but I mean, we do have some evidence that in the challenge nursery, there is that trade-off. But then we also have the pigs that make it into the finisher. They are sort of on a recovery phase. So the other thing that we also have to look at, maybe that the pigs that respond strongly to the immune challenge, they may have lower growth rate in the nursery, in the challenge nursery, but then they get over it and they actually grow faster in the finisher, right? Because there's this whole allocation theory. There's only so much energy and it has to be allocated. But another part of resource allocation is how flexible animals are in reallocating, right? So, and we've seen that in some of our PERS work, that the animals, actually the line selected for low RFI to be more efficient, you would think that it puts all its energy, it can't amount a very strong immune response. It actually mounted a very strong immune response, dealt with the disease, and then went on growing. Whereas the high RFI line, the inefficient line, didn't have as strong as an immune response, and as a result, the infections kind of lingered on, right? So this metabolic flexibility, as we call it, is another aspect that we have to look at. And but yeah, no, we haven't looked at that in great detail in these data.
Thanks.
Okay, so we'll give the floor to Maria Ballester, who has a question. She's in the room.
I have a question regarding the immunomarkers that you tell us that the correlations are opposite in the experiment about the cell-mediated and antibody-mediated immunoresponses, that you were expecting the same results that in cows. And my question is that if you have seen that there are a high proportion or the mortality is more related to respiratory diseases, maybe that the percentage was higher, these results maybe due to the specific pathogens that are circulating in the farms, and maybe are expecting, because maybe you have more peers or more, I don't know. My question is.
Yeah, that's a very good point, and we're actually now starting to separate out the mortality and treatment reasons, right? So we can specifically look at the relationships with mortality due to respiratory disease or treatments for respiratory problems, and I think that will get at your question. But yeah, we don't have those results at this point yet. So for now, we've lumped everything together and looked at, regardless of reason, we looked at mortality and treatments. Yeah.
Thank you.
Okay. And there is another question from Peter Knapp. Peter Knapp is online. Peter? Well, he raised the hand, but Peter, are you there? Yes.
Yeah, yes, I'm online, but I couldn't unmute myself. Hi, Jack. Nice to see you again.
Yeah.
This is really good. you showed us about, I think, 250 genetic correlation estimates, and very, very few of them were statistically significant due to the huge standard errors they have. And most of them were based on something like 1,000 animals. Yeah? Now, what you could do, and maybe that would be a very interesting and useful little project, is push all that data into a power equation to figure out how many data we would need to make those estimates, let us say, robust and meaningful and trustworthy, etc. Any idea? Are you going to do anything like that? Because in 5 or 10 years from now, that may very well be the most important output from this little project. Know what we need to do to make it work.
Yeah, that's a good. We haven't done that, and I hadn't thought of doing that. I mean, of course, there is the main goal from an industry perspective was to identify potential candidates, indicators that look promising, and then collect more data on those. But yeah, I think we can get a better idea of how much we can certainly use our data to understand how much data we need.
Yeah, because those potential candidates, from a negative point of view, a pessimistic point of view, that would take us back something like 15 years or so, when marker-assisted selection was supposed to save the world. And we all know what came out of that. So some kind of quantification of what kind of data do you really need to make this work in a meaningful way may be an extremely useful bit of information. I would argue.
Yeah, no, that's you're correct. Yeah,
and you seem to be sitting on that kind of data.
Yeah.
Okay, thank you.
And the other thing that I mean, there's still lots more we can get out of this data. One of the things that we are doing also is now with all these immune assays. I mean, they're all correlated with each other also, right? And so that we measure different aspects of the immune system with different types of assays. And so one of the things we're also doing is trying to get more biological understanding of which aspects of the immune system are most important when we aggregate all the data together.
Yep. Okay. And to come back 4 minutes ago, when you said reallocation may be just a thing, I would very heartily agree with that. That is what we need to focus on.
Yeah.
Okay, thank you.
Yeah, thanks.
Ricardo, switch me off, please.
We have another question from Marco Bink in the room.
Hi, Jack. This is Marco Bink. And a bit follow up on what Peter just mentioned about how many animals do we need. I had a question about, given all what you learned from this project, and to what extent is the choice of your material critical, and the way you organize it? Like you had the large white landrace crossbred, but they came from 7 different providers from different locations, etc. You mentioned that these animals may already have been stressed before starting the experiment. Can you give any recommendations or reflection about what could be done better or in the next project?
Yeah, well, first of all, I want to say, I mean, I want to emphasize that I'm part of a large team, right? And Graham was very involved and others. So I'm just the one speaking to you today. But so I didn't do all the organization. But in terms of what can be done better, I think it was important that we had multiple lines involved, animal lines, and that they were crossbred, because commercial animals are crossbred. And of course, always a challenge is to get access to those animals. And breeding companies were very eager to collaborate on this. They see it as pre-competitive research that is very important to all of them. But yeah, I mean, it's impossible to get nucleus animals, and so multiplier barrows from the Yorkshire Landrace cross were available, and so but yeah, then multipliers aren't as clean as the nucleus. So whether that has a big impact, whether what we see on these animals in terms of their immune assays also applies to the purebred nucleus animals, we don't know. If you really want to look at implementation, then it would probably have been better to get nucleus animals for the immune assays, for the indicator traits, but we need to measure the disease resilience on crossbred animals, right? So it's not easy, but you know, the things that can be done to better target the specific questions that we have in terms of developing indicator traits that can be measured on purebred nucleus animals.
Thank you.
Okay, I think there is a question in the chat. So I'm trying to recover it.
Yeah, from Chris. Chung et al. Just published a paper that indicated RNA traits can explain a large proportion of heritability in cattle traits. Can you talk about the EQTL work and any evidence that these data can explain the resilience traits? Well, we've done it at the phenotypic level. We've tried to predict the disease resilience from the gene expression work, for the gene expression data, and we used the genomic prediction machinery as well as machine learning. And it's not very, at the phenotypic level, it's not very predictive. At the genetic level, well, the genetic correlations we're finding, they do indicate that there are that the gene expression in blood at this early age does explain at least some genetic variation of disease resilience. We haven't done the calculations to exactly say, to talk about how much. The EQTL, yeah, of course, when we do the EQTL work, you know, that's regardless of the resilience data, right? We just look at analyzing, identifying QTL for gene expression. But then that may lead us to better candidate genes for disease resilience also. But we're not far enough along there yet to be able to say anything about how much that can explain.
I had also a general question, Jack. It's about the durability of this facility that you are using, because if I understand, you started in 2015, and I can imagine the amount of permissions for ethics and all this that you have endured in all this. So, I mean, it's something that probably in Europe it would be extremely difficult to realize. And I was wondering, I mean, do you have a term line, or you think that you can just continue? I mean, you will continue for long, or you will stop? Have you planned to stop in this?
Well, again, it's not me, and it's not me making the decision, but it's the test station of CDPQ, so actually the Quebec government, provincial government, and the staff there. The main person is Frédéric Fortin, and they have actually now terminated the experiment because it's expensive, and you know, we're not able to get additional funding to continue this work at this point, right? But yeah, I mean, it did involve a lot of you know, certificates, health certificates, and approvals, and which, yeah, I agree, may be more difficult in Europe, and not to say that, you know, in Canada and the US it's everything is allowed, because there was a lot of oversight and a lot of going back and forth between the staff at CDPQ, and John Harding, who's a veterinarian from the University of Saskatchewan, was very involved, and a whole team of veterinarians to put in place protocols to minimize the suffering, but still maintain a strong disease pressure so that we get data that's relevant.
Yeah, I didn't say that in the US it's easier that much. It's just that, I mean, It was
Yeah.
just an assumption from me.
Yeah.
So I don't know if there are other questions. Probably not. And in this case, Jack, thanks a
Okay.
lot again.
Okay. Thank you.
Really thanks for being with us. And I hope you will be with us tomorrow as well.